So we're delighted to have Professor Katharina Pachmann with us today. This is the Academy of Nutritional Medicine. Professor Katharina Pachmann is with us from Germany. She has been running the Maintrac laboratory there in Bayreuth for 20 years now, with a large group of staff. I think they're about 50 now. She's both a medical doctor and a research scientist, and has been conducting studies in the field of cancer, and particularly how to perform liquid biopsies of circulating cancer cells for over 2 decades, refining these techniques. And she's authored over 100 scientific publications on this technique, which are available either via our own website or the Maintrac website, which we link to from our own site. So without further ado, we look forward to your talk, Katharina, on the efficency of apoptosis, of natural agents, of these circulating tumor cells. She may call them CTCs or CCCs, circulating cancer cells. And she'll also give us an overview of therapy relevant characteristics of these cells. So thank you very much, Katharina.
Thank you very much, Gillian, for introducing me. I will not go any further in my career and will start immediately talking about apoptosis, efficiency of natural agents, and an overview of therapy relevant characteristics of circulating cancer cells. Our method is called Maintrac, and we use EpCAM as a way to find the epithelial cells among the blood cells. Why do we use EpCAM? EpCAM is the name of epithelial surface antigen of epithelial cells. EpCAM is present on the majority of carcinoma cells. It's expressed on the surface of the carcinoma cells. So for detection, there is no fixation necessary, which would be necessary for intracellular epitopes. And this fixation would prevent distinction between live and dead cells, and it can change and destroy surface epitopes. So that's why we are looking directly for the EpCAM on the circulating cells. You can imagine that on a cell, there is a molecule which transgresses the cell membrane, and it can be visualized by tagging it with a fluorescent tag. And we use antibodies which are fluorescently tagged to detect these cells. So you send the blood sample, we take 1 ml of blood and lyse the red blood cells which would interfere with our analysis. And then there is this tiny little sediment of white blood cells, and among these blood cells there are the tumor cells intermingled, and then we add this antibody. The antibody does not attach to blood cells, but only to epithelial cells. Then we add the samples to a 1696 well plate, and in every well there is the sample of 1 patient. The microscope then scans over these wells. It takes 100 pictures of each well, and then it presents us the cells in this way, and we can then look at them. And if we do a repeated analysis, we can draw a trajectory of the numbers of cells over time. There are other technologies available. The most well-known is magnetic bead enrichment. I won't go very much into this, but it's the question whether the EpCAM expression is sufficient for magnetic bead enrichment. there are microfiltration methods which assume that the tumor cells are larger than the blood cells. They are sometimes not. Or negative depletion, you take away all blood cells using this antibody against blood cells, white blood cells. But you lose a lot of cells, and then there are more methods. Then there is a method called Oncotype DX, which perhaps many of you know, and which is used by some gynecologists. Oncotype DX looks for the expression pattern from the primary tumor cells. We are looking for the tumor activity by monitoring the behavior of the cells that have reached the blood. Oncotype DX forms a risk score, and they claim that patients with a low risk score might forego chemotherapy. For us, an increase in cell numbers is a sign of tumor activity. So we do not rely on the primary tumor, but on the cells that have left this primary tumor and are present in the blood. And a decreasing number of these cells indicates that the therapy has been effective. It's not yet finally determined whether the clinical scores relate to this score. There are still studies ongoing, but we know that stable cell numbers without therapy indicate that these cells in the blood are dormant. So ours is a monitoring system, whereas Oncotype DX is a prognostic marker from the primary tumor. Very much has been talked about the circulating tDNA, the circulating tumor DNA. This is DNA which is derived from dying cells. We, in contrast, only count live cells. And it's a small fraction of the total DNA, and is influenced also by the decay of the white blood cells in the blood. We are looking only at the tumor cells, and we just don't look at the white blood cells. It's known that if the cells decay, there will be an increase in circulating DNA. In our approach, an increase in cell numbers is an indicator of renewed tumor activity. ctDNA, just make it short, can be used only in metastatic disease, whereas circulating tumor cells are detectable at all stages of disease and can be monitored also in the primary situation. And the main purpose of the circulating DNA is to detect mutations, which then can be addressed by certain drugs. We look at the tumor cells independent of their mutations and just look at their behavior. So what are these cells looking like? That's a green stained tumor cell, and adjacent is a red stained cell. In this case, it's a blood cell, and you see that there are other blood cells around which are not staining. The red staining are the lymphocytes, and the not staining are the normal granulocytes. And down here is another cell which has tiny staining. We have also counterstained our detection method of EPCAM with a molecule called cytokeratin, which is used by many others. But the disadvantage of cytokeratin is that you need to fix the cells because cytokeratin is intracellularly, and then you can't anymore distinguish between live and dead cells. So we show that cytokeratin is expressed in the same cells as EpCAM, but it needs to fix the cells, and that's what we don't want to do. Here is a gallery of cells found in one patient. And here is this important feature which we use, that we distinguish between the live cells and the dead cells. How can we determine the dead cells? If a cell is dying or going into apoptosis, the membrane becomes permeable, and a dye, which is called propidium iodide, which intercalates with the DNA in the nucleus, can enter the cell and stain the nucleus red. So cells with a red nucleus are dead cells. Cells without a nucleus are the living cells, which we are looking for. And what we do is then we draw a trajectory of the cell numbers over time. You see here, it's about 1 year. And what we saw was the cell numbers decreased over time. And indeed, this was in spite of an increase in serum PSA, but the patient is still in complete remission. So the PSA was less less effective or less telling than our cell numbers. On the other hand, this is a patient with a colon cancer. He had curcumin during this time here, and his cell numbers first decreased during this treatment, but after stopping, cell numbers increased again, and he suffered relapse here. After the relapse was operated on, we can see that the cell numbers did decrease. So we have now lumped together all our results of circulating tumor cells. We call them circulating epithelial tumor cells. And we have, during 15 years of follow-up, we could identify 439 cases where we had clinical data available. And this is to tell that it's very important for us to draw a concrete to tell you how the cells respond to the therapy if we get clinical data from you so we can correlate them. In these primary tumors, 276 had cell numbers decreasing and 173 increasing cell numbers. In the patients with increasing cell numbers, the mean relapse-free survival time was already reached at 1.5 years. Patients with decreasing cell numbers had a mean relapse-free time of 8 years. The difference is highly significant, and those with increasing cell numbers had an 8.8 higher relapse ratio. And this is the curve. So this is Kaplan-Meier curve. At this time point 0, all are alive. And those with the increasing cell numbers then suffered relapse, and every tick down is 1 relapse. And the mean was reached at 1.5 years, whereas those with decreasing cell numbers are plotted here. And you see the mean relapse-free survival was more than 8 years. So those with decreasing cell numbers have a very good survival. In contrast, we could test the same trajectories also on metastatic tumors. So it's not neither restricted to primary nor metastatic tumors. We can test all sorts the stages of tumor. Here we had 341 cases with clinical data available. And you can see in this case with the metastatic tumors, it was lower number of cases with decreasing cell numbers and a higher number of cases with increasing cell numbers, which is due to the less favorable prognosis of these patients. Here, those with the decreasing numbers had even shorter mean survival time of 0.7 years. But if cell numbers were decreasing, in spite of these patients having already had metastatic tumor, when they had decreasing cell numbers, their mean progression-free survival time was still 4 years. And again, this is highly clinically significant with a hazard ratio of 4.8. And here is again the survival curves. Those with increasing cell numbers have a relatively rapid decrease, relatively rapid relapse, and those with decreasing cell numbers have a good survival. We have not yet analyzed all these patients, whether they have been treated and with what therapies they have been treated. This is all still in our data, and we hope to be able to provide more data to you soon. So I think no other lab has a comparable flexibility. We test not only conventional agents, but also natural agents for their ability to kill the cells. And it's always the patient's own cancer cells. It's not a median of a lot of patients. You can send in your own selection of agents, or you can select them from our list of suggestions. And it's important that some of these agents work quite different whether they are given as an infusion or an oral supplement. So it's important for us to know how you want to give them. We can also test mixtures of agents, and you can choose the combination. So how do we test these agents for their ability to kill the cells? We have called it chemosensitivity testing and published it in 2013. And what we do is we look at one and the same cell over time with the addition of a drug. And as you can see, over time, this cell, which first looks quite happy, dies over time. The same is true for this drug and for the marphosphamide, which is cyclophosphamide. This is just to show that with conventional drugs, we can see the effect, and then we draw conclusions for natural agents. And this is just to show you how these cells die over time. And if you look at this cell, it has been looking happy before, but it gets more and more red. So the membrane becomes more and more permeable. And this cell down here remains alive. So we can distinguish between the cells that die and those that remain alive with the same agent which has been added to this suspension. We then looked for conventional agents, how they look clinically. And what we saw was that cells that are sensitive to certain drugs, this is a drug combination, in our in vitro assay, patients survive much better relapse-free. It's statistically highly significant as compared to patients whose cells are resistant to these agents, which do not die upon addition of these agents. And the difference, again, is highly statistically significant. Here is a case of a patient. She had an ovarian carcinoma. She got the conventional treatment. And what we saw was that during this half year of conventional treatment, her cell numbers increased all the time. And only at the end of the treatment, they performed imaging, and they found a progress. So she had to suffer all the time the disadvantages of this treatment, and it didn't help her. Then they switched to another agent, which we had tested to be effective all the time. And we had tested the two given drugs all the time as being very low or ineffective. And when they switched, her cell numbers went down, her disease stabilized, but you can't give these drugs for a long time. So if they had switched to this drug already earlier, maybe she would have been faring better. This is also one other patient, just to show she was treated with different conventional drugs. Her cell numbers first responded, but the sensitivity to these drugs decreased all the time. And in the end, indeed, she increased in cell numbers again. So the sensitivity to different agents can differ over time, and they do so mainly upon treatment. But we can also use natural agents. And we have here been looking for a series of patients, and we have been recording the sensitivity, so how many cells, what a percentage of cells was killed by these drugs. This is first a conventional drug at 50%. Okay, there are about half of the patients are sensitive and half are resistant. This is somewhat better, but doxorubicin is also much more cytotoxic and toxic to the patient. So the improved effectivity on the tumor cells is brought by a higher cytotoxic or toxic agent. This is vitamin C. And as you can see, there are a lot of patients who respond very well to this vitamin C, but there are also patients who do not or almost not respond to vitamin C. So if we test in advance, we can tell you this patient will respond to vitamin C. And moreover, as I will show later, you can also increase the concentration of the drug. This is artesunate, and you can see very many, about 70% of the patients respond with a good response to this drug, to this substance. And the best outcome was for us with curcumin, where many patients, almost all patients, respond well to this substance. You can, as I mentioned, choose among different substances which we suggest, but you can also ask for other substances. You can call us if we have the substances available. These are certainly available, but we have other natural substances also available. And you can do combination testing. You can combine your drugs. So this is how we present our results. Here is the anamnesis, the cause of disease, and here is the number in per ml, and we calculate them in the whole blood system too. And we also tell you whether there are already cell fragments present, because this might tell you whether the therapy has been working. In addition, like here, we tell you what the results of the tested substances is. And as you can see here, this a patient responded well to vitamin C, not so well to another provided himself. Here, another patient, cell number, and then here the different substances. And you can see here that the natural substances in this patient were better than the conventional substances. This is not rare. We can see this quite often, that a range of natural substances are more effective against the circulating tumor cells. What we also can do and what you can request is a concentration dependent report of the different drugs or of the different substances. We always do this concentration dependent analysis because only if it's concentration dependent we believe that it's really working. And you can see that the curves can be quite different. So this starts at the 0.1-fold concentration, very low, and comes up very high. Curcumin didn't work with the other concentrations in this patient, but we have here another patient where we could show that already at the 0.1-fold concentration, there was a good response, a good cytotoxic response, and that's even further increases. Here is a combination, and here you can see that sometimes the combination is not better than the single substances, but in this here the combination was highly effective. Another combination, curcumin and hypericin, which showed to be very effective. Here you see a trajectory of cell numbers over time, and here, although this was varying, you see that the cell numbers nicely decline upon treatment according to what we suggest. And furthermore, that's the same patient. After this decline, the patient had a stable number of cells over more than 1 year tested, and still has upon treatment with, in this case, curcumin and vitamin C. And the patient was also in complete remission. Here again, hypericin at different concentrations, and here you can also see it starts very low, but then it comes up. So it's very much dose dependent how effective the drugs are. This number is the concentration which we usually use for, and which are reported to us to be used in the patient in the infusion. As mentioned before, the effectiveness can change over time, and this was the case here, where capecitabine, which is a form of 5-FU, was very effective in 2017, but was ineffective 2 years later. So but together with curcumin, it again was effective. So it's really worthwhile not to test for only once, but after the patient has been treated, and you want to treat the patient again, to retest. For the treatment of the patients, it's also important to know characteristics of the cells which are relevant to therapy. We can look for additional markers apart from EFCAM with the use of different colored fluorochromes at the same time, and then tell you whether on these cells certain markers are expressed. We have different typical characteristics. Some of them are the hormone receptors, growth factor receptors, and I'll go through it now. So, hormone receptor. The estrogen receptor is very much important if the patient is supposed to be treated with endocrine treatment. I here mentioned just the conventional endocrine substances, but there are also natural substances available which you might consider. So it's important whether the cells carry this receptor, and the progesterone receptor is also complementary to the estrogen receptor, and the androgen receptor now becomes obviously important not only in prostate cancer but also in triple negative breast cancer. Triple negative means they do not have the estrogen receptor, they do not have the progesterone receptor, and they do not have HER2, which I will come to later. This is a typical staining for the estrogen receptor on individual cells. So these cells are EFCAM positive, they are from the tumor, and they are estrogen receptor positive. And this is a patient where the tumor had been shown to be negative for estrogen and progesterone. But we tested them, the circulating cells, to be positive for these receptors, and indeed the cell numbers remained very nicely stable over a long time. That's just the time when we tested it. So if they are treated correctly, they can be stable cell numbers or decreasing cell numbers even after years after the tumor has been removed. So we would like to emphasize that in the first report on the primary tumor, the cells were declared to be hormone receptor negative, but in the circulation, they showed to be positive. And this occurs not rarely. It's about 25% of patients which change their receptors from the primary tumor to the circulating cells. But on the other hand, often the circulating cells maybe have the same characteristics as the primary tumor. Other receptors are so-called growth factor receptors. There is the most well-known is HER2, and HER2 was seen as a negative prognostic factor as long as there was no treatment against this factor. But now, there is an anti-HER2 antibodies available, and the prognosis has changed. EGF receptor is relevant in lung cancer and for application of gefitinib or erlotinib. Then there is the growth, the factor for growth of vessels, and this is also important to know. And the insulin growth factor receptor, where there is quite a lot of therapies available because these cells respond to insulin, and you can either reduce the concentration of the therapeutic agents or get onto this receptor by other ways. And c-KIT is also a growth factor receptor, which is typically expressed on stem cells. These, the presence of these receptors is frequently determined by fish analysis, and I'll show examples of this immediately. So this is a EpCAM positive cell, and it should have only 2 signals of HER2 because it has the signal from the father and from the mother, but this cell shows a lot of signals, as this does. It's not really known how this occurs, but it's known that these the cells can amplify, repeatedly read these gene sequences, and then have a lot of these sequences present, and this means also they can express these growth factor receptors at a higher concentration on their surface, which means they can respond better to growth factors. And we have to block these receptors to make the cells insensitive to growth factors. One other type is PD-L1, which has been talked about quite a lot. PD-L1 expression can be detected on the circulating tumor cells, and it's known that these PD-L1 receptors can be inhibited by some of the substances. So it's called immune modulatory therapy. Immune modulatory because the tumor cells have the capacity to silence the immune T lymphocytes and to tell them, well, I'm not dangerous, just leave me alone. And this is made by the PD-L1 and PD-L2 and other so-called checkpoint inhibitors. We can test for them, and as you can see, again, an epithelial cell, PD-L1 in this cell, and the merged pictures, or PD-L2 in these cells. So we can test for these receptors and tell the treating physician this patient has PD-L1 positive cells. We can also detect these PD-L1 by, again, fish, because it's known that in order to express increased PD-L1, again, the genes can be amplified, and this leads then to an increased expression of PD-L1. And this we have shown here. The more amplificates there are present in a cell, the higher is the expression of PD-L1 on the cells. We have shown that this can be increased in the metastatic situation, but also by radiation. Radiation can induce PD-L1 expression. And here is depicted how this works. The tumor cell tells the T cell, don't touch me. And this has to be interrupted by the respective therapy. Here we have one case of a melanoma. He had high PD-L1 expression. Cell numbers went up steeply, and then he got a treatment. I'm sorry, it's not visible here. And his cell numbers went down, but after a while, his PD-L1 expression came up again. This is another patient, a breast cancer patient. She received, she was HER2 positive because so she received this treatment, but also here a treatment against PD-L1. Her PD-L1 expression went down, although her cell numbers went up, but with the next treatment, cell numbers came down again, and after a while, you see PD-L1 expression starts coming up again. So this is an effective treatment, but many of the patients, again, suffer relapse. I was asked also to talk about Ki67, which is tells us if the cells express Ki67, they are in aggressive growth if the Ki67 is higher than 30%. And we again could see show that in this case with a high Ki67 expression, vitamin C was very effective. We can also test for P53. In this patient, it was a high expression of P53. P53 is a protein which usually is expressed in the cells, but degraded rapidly, so you usually can't find it. But if it's mutated, it will be detectable. And I know about a Chinese group who treat patients for P53. So if somebody wants to know, I can look it up later. What we also can do, and what is a sign of more aggressive growth, is clonal expanding circulating tumor cells. Not all tumor cells can do that, and we call them tumor spheres. And what do they look like? It's small spheres, it's clumped together cells which come from one cell which has been dividing, and these spheres grow over time. Sometimes the mother cell is not positive for EpCAM, but as you can see, the spheres are positive for EpCAM. So they are derived from epithelial cells. How do we proceed? We proceed in the same way as for the circulating single cells. Again, here we have the sediment. Then we put it into growth medium, which prefers growth of epithelial cells. And then we find some of these circulating epithelial cells have grown into these spheres. What do these spheres look like? They have a typical pattern of so-called stem cell markers. They are positive for CD44, negative or low for CD24, and they are positive for an enzyme which is called aldehyde dehydrogenase, and they do express these markers. These tumor spheres can also be tested for their sensitivity to different growth, to different drugs, and this is curcumin, which had been applied to these three spheres, and you can see how the spheres die over time. Here is a comparison. This was a control with no substance in it, These are conventional substances like taxanes. They don't touch the spheres, but salinomycin, you can see how the spheres from the beginning start to die. Sometimes some of the cells survive, and curcumin also is effective in tumor spheres. In these cells, which have been grown from one single cell, and show that the tumor is higher aggressive. And this is a comparison. Single cells are often very effectively killed by these taxanes, but they don't touch the spheres. But salinomycin and curcumin may be very effective. But sorry enough, sometimes this doesn't work. In this case, salinomycin was active against the single cells, but not against the spheres. So, in conclusion, say we can identify the circulating tumor cells, we call them circulating epithelial tumor cells, and can characterize them, and not only in metastasized situations, but also in the primary tumor, where patients may be cured. Our analysis is quantitative, and we can test in advance the efficacy of a medication, and then we can, in addition, look for the cells, whether they really die. And we think that in the future, and already now, treatment decisions can be made with the help of our method. We are fully accredited, and we are now applying for the CE certification in Europe. That's the shipping device which we can send to you, because it keeps the temperature quite stable. Here, how you add the samples into this shipping box, and that's our laboratory, and the samples should arrive between 48 and 72 hours at room temperature at our lab, and the results you will get 5 days after we have received the sample. Thank you for your attention.
Thank you very much, Professor Pachmann. That was fascinating, and a lot of extra information there that we haven't had before on, for example, the comparison with other test methodologies, as well as the therapy relevant characteristics. Just to mention to everyone here, there are other recordings of Professor Pachmann that you'll find on our website under events, and then webinars, or otherwise please just ask us if you'd like the actual voice recordings. We do have those too, and we've had events where Professor Pachmann has come over as well. The recordings are available on the website too. Just to mention also that AONM, the Academy of Nutritional Medicine, is the hub for these tests in the UK. So all you need to do is go to the website, click on the tab Maintrac, and all the information is there, or simply give the helpline a call and the kit will be sent out to you. And we have lists of the different laboratories or clinics available where blood can be drawn around the country. So just to mention that we do have a little time for questions. Professor Pachmann, if that's all right. I know it's really only an hour, but we have about 11 minutes or perhaps a bit longer for questions, and some were sent in kindly in advance. So I'll begin with a couple here. Is there a difference in terminology between cancer and tumor? Sometimes CCCs is used as a term, circulating cancer cells, and sometimes circulating tumor cells. Is there actually a difference?
No, there is not really a difference. In German, a tumor means just an increase in size of a tissue, but it's used usually for malignant tumors. So cancer is one sort of malignant tumor. It's the epithelial tumors, and there are other tumors like rhabdomyosarcoma, leiomyosarcoma. So sarcomas are a different sort of tumors, but they sometimes also have epithelial antigens on their surface, and they are rare. The frequent ones are the cancers, which are of epithelial origin.
Thank you. And this same attendee asked, I was made aware that on some occasions CTCs are not visible, and the term hiding was used to explain this. I understand there's a proper term for this, but if cells can hide, how do we know that some of the cells in any host are not in this sort of dormant or hidden phase?
Yeah.
And that perhaps the cell count is a little different to the one being presented at a particular time?
Yeah, that's true. And indeed, if they are hiding, we won't be able to see them. But if they are aggressive, we have seen that we can make them grow into spheres. And there are patients who have very few EpCAM-positive cells, but a high number of tumor spheres. And then again, we can detect these hidden, very aggressive cells and can determine their number. And the patients with metastatic disease or in the course of the disease can increase in their numbers of tumor spheres.
Yeah, so this is, go ahead, please.
Yeah, it's the problem is that we need about 3 weeks to have them grow. So they are not immediately available.
So this is something that's best discussed between the therapist and you, perhaps. And yes, we do often get questions from therapists about what the best types of tests are to do, and we do put them in touch with you. You're often very kindly available for these kinds of questions. So the important thing I think to mention is that Professor Pachmann is always there also to discuss the results that come through. Generally, AONM has a look at them first, and sometimes they're answers that we're able to provide, but if not, then we do direct the questions straight to you. Another patient here asks, what's the prognosis of a patient with a Ki-67 that's over 50? And how many years before such proliferation is manifested in imaging?
Okay, the first question is, they are at a higher risk, but the Ki-67, it depends whether the patient refers to the primary tumor tissue where the Ki-67 is determined, or for the Ki-67 which we determine on circulating cells. So if, for example, the primary tumor had a high Ki-67 and is removed completely, and the cells which are in the circulation are low Ki-67, then the prognosis is much more favorable as the one who have circulating Ki-67 positive, highly Ki-67 positive cells in the circulation. And again, we can look whether the respective therapy reduces this activity of Ki-67.
I think you showed a case where both curcumin and vitamin C seem to be effective.
Yes.
Yeah.
Yes.
And then the same patient asks, in your experience, have you seen a big decline of Ki-67 index in patients, and what agents attributed to this decline? Well, I mean, you've already presented that one case, but have you got any other suggestions?
Well, I would actually need to look up our results. I can't just tell now but I would be able to look for all our results with respect to TI67 and draw a conclusion.
That would be very helpful, and maybe we could feature that in a further presentation we have with you, because I suspect this is a patient who's had that measured and is wondering which substances to test against it. Another patient here, or at least attendee, asks, the tests measure the effect of agents used directly on the cancer cells in vitro. To what extent can we expect them to have the same action in vivo, particularly when passing through the digestive system? Do you have any information?
When passing through the digestive system, I can't really tell. I know that curcumin, for example, is only 1% of what you eat will get into the circulation. That's the problem with curcumin, because it's only it's bound to fatty acids, and but there is soluble curcumin available, and we can use these, and then we can look for the concentration of such a substance in blood and whether the cells respond. What we have done is we have accumulated now conventional drugs and looked for their effectivity in vitro and in vivo. I haven't shown these pictures, but the higher the in vitro cytotoxicity was, the better was the reduction of circulating cells, and this also correlated with clinical results. And we are going, or we will do the same for natural agents, but I don't have these results available yet. But it seems that in vitro testing quite well reflects the in vivo sensitivity, and in addition, with respect to conventional drugs, if they don't work in vitro, there's a low possibility that they will work in the body. So you can then possibly avoid treatment with certain drugs if they don't work in our in vitro assay.
Thank you. I'll come on now to those that have been asked online. Can a test be used when a man is already using drugs for prostate cancer, and one is a hormone drug that he's been using for many years, by including the information, or would this patient have to stop the drugs before testing?
No, it has not to be stopped, but it has to be done repeatedly. So we need to see whether the cell numbers remain stable or even decline under this hormone treatment. And we do that quite a lot, and we see very good results. If the hormone treatment works, cell numbers remain quite stable or even decline over time, and the man remain without progression of the disease. So it's not necessary to stop. We can see the function of the treatment directly, but we have to do it repeatedly. It's not one single number of cells which can tell the results.
And what period of time would you suggest? Once every 3 months or every 6 months?
With the hormonal treatment, once every 3 months in the beginning, and then you can extend it to once in 6 months. But what we also have seen, I would like to emphasize that, and we are going to do a study on that, that is for breast cancer patients, at some time they just stop their hormonal treatment. And then we can look at if in many cases, cell numbers remain nicely stable even if you stop the hormonal treatment. But if the cell numbers start increasing, then we would recommend to restart treatment.
Yeah, thank you. And if it's a matter of chemotherapy, I know hormone treatment is a bit different because you're blocking the cell receptors.
Yeah.
But in the case of chemotherapy, how long should you wait, ideally, before doing a test? Does it depend on the type of chemotherapy? Does one have to look at the half-life?
No, rather on the response you want to have. So if you want to know, has the chemotherapy been effective, then you can look immediately after the end of chemotherapy whether the cell numbers have been down, or often they're in this. We can't see any cells anymore. But then you have to look at the cells after a while, like 6 weeks, 8 weeks. And if the cell numbers come up again, this is a sign that there should be an additional treatment. And this can be naturopathic also.
Thank you. I know that you often state the number of cell fragments that have been found, and that can indicate the effectiveness of the therapy too. But so is there ever any benefit in waiting for like 1 or 2 weeks after chemotherapy until those cell fragments have cleared before you check what the status is? Or do you always suggest testing right afterwards?
No, it depends on what you want to know. If you want to know that after chemotherapy, whether there are still cells coming up again or still present after a while, you can test only after 6 weeks. But if you want to see whether the immediate chemotherapy has been effective, then you can test immediately after the end of chemotherapy, and you will see whether there are still cells left, an indicator that the therapy has not been that effective, or whether the cells have disappeared, a sign that the chemotherapy has been effective.
Thank you. Another question, is there any guidance available on which agents would be best to test for certain cancers?
They are not different for different cancers. They are different for different patients. But no, we don't have any indications that, for example, colon cancer is especially with naturopathic agents, is especially sensitive or resistant. So you have to test it on the individual patient.
Okay. But maybe it would be possible at some point to get together a list from your experience, because you've been doing this for so long, of at least the types of agents that have been tested. And as you had with the curcumin and the artesunate, you know, the ones that are most likely to be effective, just so that patients and therapists can target their testing. That might be something we can do over time, do you think?
Yes, we should do that, Gillian. Yeah. Yeah.
That would be super, I think. Let's think about doing that, and then we can have a further presentation on that. So thank you for that question. And another here, it's actually a follow-up of the same question. Once a sample is provided, can testing be done with successive agents if initial agents are not effective? I don't think so with that same sample, because you need live cells, right?
Yeah, and the cells die over time, and just simply to test for a certain agent, it takes 1 day. So the cells will be less viable after the several days. So if you want to know immediately, you can call, and then we'll try to answer, and then it might be possible to add additional agents.
Yeah, but you still won't get the full effect, because, as you say, some of the cells will already be dead, and so you won't be able to work out that how much of the 100% has the agent managed to kill, which is your ideal ratio, isn't it?
That's correct. Yeah.
Okay. Well, Thank you so much. There are a few more questions, but I'll either send those in to you, or maybe we can feed those into the next session that we have with you. Thank you so much. That was
Thank you very much.
really
If they are urgent, you just mail them to me and I'll try to answer.
Thank you, will do.
Thank you very much. Thank you.
Thank you to everybody who's been on with us here. Many thanks. Do mail us in your questions if you have further ones that you'd like to ask Professor Pachmann using the email info@aonm.org. And the Academy will be holding further webinars in November and December. One round will be on the PANDAS, that's the neuropsychiatric conditions that can be triggered by infections. We'll have specialists from the States talking about those. And we'll also have Chris Willems back again from Cancer Active to answer your questions, because there were so many from the time he talked in our cancer round previously. And so we'll be sending out emails about those. Thank you very much for attending. Thank you again, Professor Pachmann.
Thank you for having me.
Thank
Goodbye.
you. Bye.